So we are now on the second part of Module 8, wherein we will be talking about optical instrumentation. Okay, so basically, optical devices have the following components. You have your light source, you have your wavelength selector, sample holder, your transducer or detector, and of course the signal processor and layout. Now shown here in this figure are the different ways these components are arranged. Alright, so this shows you the optical pathway, no? Yung dinadaanan na ilaw across the equipment. So for absorption spectroscopy, so ano yung mga absorption spectroscopy? You have your UV vis, IR spectroscopy, okay? They follow what I'd like to call a linear pathway, no? So you have here the light source, you have the wavelength selector, wherein you can produce monochromatic light, okay? Then the monochromatic light passes through the sample and the detector so that you can measure the transmittance. Then by computation, you can measure the absorbance. Now for fluorescence and scattering spectroscopy, what we have is some sort of a perpendicular optical pathway, no? So notice that for fluorescence or scattering experiments, you have two wavelength selectors. Now for fluorescence, the first wavelength selector is what we refer to as the excitation wavelength selector. So this controls, no, the wavelength used to excite your sample, no, to induce fluorescence. The second wavelength selector is your emission wavelength selector, which allows you to filter out, no, the wavelength, okay, of the emitted radiation, no, from your sample. Okay, then finally, this is for emission spectroscopy. Notice that there is no light source because for emission spectroscopy, your the sample itself is the one that is emitting radiation and not due to excitation by a light source, no, but rather via flame, plasma, or electrical spark. Chemiluminescence, no, spectroscopy will also use this kind of setup, no, and the excitation is brought about by a chemical reaction. So first, let us talk about the different radiation sources, okay? So first, you have your continuous source. A continuous source is a radiation source that emits radiation over a wide band of wavelengths. And examples of this are your the deuterium lamp, which is a UV source. You have your arc lamp and tungsten lamp. Okay, tungsten lamp is a commonly used visible light source. So for UV vis spectrophotometers, they both contain the deuterium lamp for the UV and tungsten lamp for the visible and near IR radiation. So basically, if you're using a continuous source, okay, you use a wavelength selector to choose which of the emitted wavelengths, no, from this light source yung gagamitin mo, no, for interaction with your sample. Next, you have your line sources, no. Now for line sources, they emit very discrete bands of radiation, meaning to say they don't emit radiation over a wide range of wavelengths, but rather mga certain regions lang, no, or certain values of the wavelength lang. One example siguro na mas ma-visualize inyo would be your lasers, no? If you have a red laser, no, kunwari ito, no, you have a red laser, it's colored red because the laser is emitting the particular wavelength for red. Alright? Common examples of line sources would be your hollow cathode lamps, okay, or discharge lamps used for atomic absorption spectroscopy. Then lastly, you have your pulsed sources. No? So basically, pulse sources can also be line or continuous source. Ang difference lang is that you are emitting radiation in bursts. Alright, so parang nag-blink. An example of this would be the xenon pulse lamp, which is used in yung microplate readers, no? Yung ELISA reader ninyo sa lab. Alright, so these are your radiation sources. Next, you have your wavelength selectors. So basically, again, you use this to produce monochromatic radiation, especially when you're using continuous light source. Okay, so first you have your filters. No? So basically, they operate by absorbing all but a restricted band or radiation from a continuous source. Siguro isang example nito would be yung cellophane, no? Which you usually use when you have classroom plays, no? Or role plays. So if you want to produce red light, no? Ang ginagawa ninyo, you attach a red cellophane, no? Doon sa ilaw sa classroom. Okay? So basically, there are two types of filters. You have your absorption and you have your interference filters. And you can read more of these textbooks in here. Then next, which is more commonly used now, is your monochromators. No? So monochromators are based on a dispersing element, no? Which is either a prism or a grating. But the grating is now used, no? Kasi mas simple yung design. No? So for the monochromator, ito yung kanyang construction. So basically, you have an entrance slit where the light from the radiation source comes in, a collimating mirror that focuses the radiation on a diffraction grating. Now, once the light, no, the radiation hits the diffraction grating, it disperses or splits into its component wavelengths. So the component wavelengths are then focused back towards an exit slit. Okay? Now, the angle of the diffraction grating or the prism is adjusted by a motor so that you can control which of the dispersed wavelength you would like to allow to exit the monochromator. No? So this is how monochromators work. Now, for filters, these are non-dispersive. Okay? Yun yung difference nila. No? Whereas if you're using monochromators, you have a dispersive instrument. Okay? The name, the jargon used for different optical devices actually depends, no, on whether they use filters or monochromators. Okay? If they are using filters, you call them photometers. Okay? So photometer, no. Example, you have your flame photometer, no. If you did your OJT sa isang soil laboratory, okay, or parang plant research laboratory, they would still have that kind of instrument, no, for analysis ng mga potassium, sodium, lithium nutrients, no, dun sa plant or soil, no. So flame photometers uses filter. Okay? But if you refer to an instrument as a spectrophotometer, alright, or spectrometer, okay, you have there a dispersive type of wavelength selector. Next, you have your sample container. Okay, so ito yung maglalaman ng sample natin. A rule of thumb for sample containers and even the other optical components nung equipment ninyo, alright, so meaning to say the mirrors, the lenses, no? So remember you're manipulating EMR here, okay, is that they should be invisible with respect to the region, no, EMR region that you are using, no. So if you're doing UV spectroscopy, kailangan the sample containers and the optical materials do not absorb, no, UV radiation. Alright, kasi remember, no, the basis of measurement for optical spectroscopy, no, is yung measurement of the transmitted radiation, especially for absorption spectroscopy, no? And another reason for that is that you don't want to reduce the intensity, okay, of the incident radiation due to absorption by the other components, okay. Importante yung intensity of the incident radiation, no, and because it contributes to the overall sensitivity of the analysis, okay. So examples of mga sample containers, you're familiar with this. You have your cells or cuvettes, you call them cuvettes. So these cuvettes are available, no, at the in different shapes and sizes. As I mentioned earlier, for example, in UV spectroscopy, the diameter of your cuvette, okay, determines the B term in your Beer-Lambert's law. Alright, so if you're analyzing dilute samples, no, that you cannot anymore pre-concentrate, then one technique is to use a wider cuvette, no, wider cuvette or wider sample cell, no? And this will give you larger absorbance response. Okay, usually you want to increase the response, no, because you want to improve the sensitivity of your method. For UVs again, no, if you remember, there are three types of cuvette, no. Merong gawa sa plastic, made from glass, or made from quartz. Okay, if you're doing visible spectroscopy, you can use either of the three, no? Pwede yon, because all of them do not absorb visible radiation. But it's different if you're doing UV spectroscopy, no? Because for UV spectroscopy, normal glass or plastic will absorb UV radiation, no? So this is the case where you have to use exclusively quartz cuvettes. Okay? Yan, sabi pa natin, no? So this shows you the relative regions, no, of the following materials wherein they are transmissive towards that particular radiation, no? So for IR spectroscopy, if you remember, we were not using cuvettes here, but rather we are using salts, no? More commonly potassium bromide. Okay, because potassium bromide, as you can see in this diagram, alright, is transmissive, no, towards IR radiation. Okay, now for double beam UV instrumentation, you are using a pair of cuvettes, no, and these paired cuvettes are matched and calibrated, meaning to say that their optical properties, na yung parang refractivity, reflectivity, etc. nila, okay, are the same. Okay, and it's important for double beam instrumentation because the assumption there is you are just subtracting, di ba, yung reading between the reference and the sample cells, no? So kung hindi match yung inyong cuvettes doon, okay, merong contribution yung difference ng cuvettes with respect to their optical properties.